The first step before starting with samples manipulation will be to clean work surfaces and the material to be used. Pipettes, waste boxes, tips boxes, and so on. Diluted bleach 1% is recommended for cleaning all work surfaces as well as material. Any commercial reagent which destroys DNA is also an option for cleaning. After finishing with the cleaning step, it is highly recommended to switch on the UV light for at least 10 minutes. Once this time is over, one can start processing samples. All kinds of samples must be manipulated under a hood in a different room from where PCR and post-PCR steps will be performed. Take swab samples out of the refrigerator and place them inside the hood. Prepare Eppendorf tubes according to the number of samples that are going to be processed and label them. There must be a tube for a blank in each run, containing all reagents used along the process apart from DNA. Pipette 400 microliters of PBS in each Eppendorf tube carefully. It is important to manipulate Eppendorf tubes without touching the inside part of the cup. Take the swab carefully and introduce it inside its corresponding Eppendorf tube. Press the swab against the wall of the Eppendorf tube in order to allow cells to detach from the swab. Take the swab out and close the Eppendorf tube. Remove gloves and throw them away in order to avoid contamination between samples. Take the following sample and proceed in the same way as previously. Then, centrifuge the samples at 2000 RPM for 1 to 2 minutes. These cells would be ready to continue with the PCR step. Place empty Eppendorf tubes to manipulate liquid-based cytology samples. Check if there is pellet in the liquid-based cytology vials. Carefully open the vial and pipette between 150 to 200 microliters of the cell suspension. Transfer the volume into the Eppendorf tubes, discard the tip and close the tube and then the vial. Remove gloves and throw them away in order to avoid contamination between samples. Take the following sample and proceed in the same way, as many times as liquid-based cytology samples are going to be processed. Centrifuge Eppendorf tubes at 2000 rpm for 1 to 2 minutes. Take them out of the centrifuge and check if the pellet is in the bottom. Carefully remove supernatant of all samples with a disposable pipette. Change gloves to repeat the process. Wash the pellet with 400 microliters of PBS by pipetting up and down several times. Change gloves between samples and repeat this process as many times as samples must be processed. Centrifuge samples at 2000 rpm for 1 to 2 minutes. Take the tubes out of the centrifuge and check if there is pellet. Then remove supernatant. Check all tubes to see the pellet and then resuspend each one with 25 to 50 microliters of PBS. Mix by pipetting up and down till getting a white suspension. These cells would be ready to continue with the PCR step. These samples can be stored at 4 degrees Celsius till performing the PCR, no longer than 5 days. For longer time, it is recommended to keep cells at -20 degrees Celsius. For paraffin-embedded tissue samples, several cuts of 5 micras are recommended for processing. Prepare tubes and label them. Depending on the size of the tissue, take between 2 to 3 sections with a forceps and place them inside the Eppendorf tubes. Forceps must be cleaned between samples in order to avoid contamination between samples. Add 400 microliters of mineral oil. Tissue sections must be covered with the mineral oil. Then heat the tubes at 95 degrees Celsius for 2 minutes. In this step, paraffin will melt and mix with the mineral oil. Centrifuge samples at 2000 RPM for 1 to 2 minutes. Remove mineral oil very carefully. A certain amount of mineral oil can remain in the tubes to avoid removing part of the sample. In any case, the remaining mineral oil will not interfere in the following steps of the process. Repeat the former step as many times as samples must be processed. Add 60 microliters of extraction buffer into each sample and mix it well, pipetting up and down. Then add 1 microliter of DNA release solution and mix it by pipetting and shaking with the fingers. Place the tubes in a thermocycler, first at 60 degrees Celsius for 30 minutes, followed by 90 degrees Celsius for 10 minutes. After this, centrifuge tubes at 2000 RPM for 1 to 2 minutes. Samples would be ready for PCR step. PCR step must be done under a different hood where samples have been prepared. Before starting with the PCR step, all work surfaces must be cleaned with diluted bleach or any other commercial reagent that destroys DNA. Any material, pipettes, racks, tips and so on to be used during the process must be cleaned too. Prepare PCR tubes on a rack. Reagents for PCR must be unfrozen. The HPV direct flow chip kit contains a vial with a hot start polymerase enzyme and a vial with the mix for PCR. For preparing, pipette 30 microliters of the polymerase enzyme tube and mix it with the whole volume of mix for PCR tube. Mix it well by shaking. Then aliquot the volume, pipetting 54 microliters in each PCR tube previously prepared. Once these tubes are ready, they can be stored at 4 degrees Celsius during 5 days or at -20 degrees Celsius for a month. If PCR is going to be performed, keep the tubes at room temperature and proceed by adding samples. All kinds of samples, swabs, liquid-based cytology and paraffin sections are prepared in the same way. From each sample, pipette 6 microliters into its corresponding PCR tube according to the number of samples that are going to be analysed. In the case of paraffin samples, take care with the remaining mineral oil. In order to avoid pipetting it, one must press the pipette before introducing it inside the tube. Then go through the mineral oil layer and take 6 microliters of the supernatant and place them inside the PCR tube. The last sample must be the blank, in order to control any possibility of contamination during the previous process. Samples can be stored at 4 degrees Celsius till having results of the run. Place PCR tubes in the thermocycler and run the PCR for amplification according to the protocol. Once the amplification has been performed, take PCR tubes out and proceed with the automated hybridization process. Bring all hybridization reagents at room temperature. Place them in their correct position according to the diagram displayed in the eBrid instrument. Then, place the HPV chips in the holder plate and fix them. Remove the cups of the reagents already placed in the eBrid. Place the amplified PCR tubes following the diagram displayed in the instrument. Cover amplified PCR tubes as well as HPV chips with their corresponding cover plates. Close the lid of the instrument and press the Start button. eBrid will start working following the protocol established. Once eBRID has finished, results can be analysed in the computer and a report can be printed.